We are going to be focusing on topic 9, which is, will we ever find a cure for cancer in animals and plants? And so we will be covering chapter 8 with this, and that is going to cover the following ideas: explaining how cells divide to give rise to identical cells, identifying the role of mitosis, meiosis, and fertilization in the human life cycle. We're going to define apoptosis, describe the steps of DNA replication, and the functions of enzymes in each step. And we're going to start with explaining what features of DNA allow two DNA molecules to form into one. And so all cells divide. That's a basic constitution for life. No living organisms can reproduce without cell division. So for unicellular organisms, things such as amoebas, they divide in order to reproduce. Now, cell division for multicellular organisms are there for a multitude of things, but essentially it's there to help with the continuous supply of replacement cells. Now, when we looked at the sexual life cycle of organisms, eukaryotic cells divide by mitosis. So when we look at that, you can look and start at adult humans. And for adult humans, you'll notice that we make what are called we have primary sexual characteristics. And that is the formation of gametes. And I think about like sex is a game. And basically, if you look at these, gametes are sex cells. So that's how I'm able to remember what they are. So there are two examples in the human species of gametes, and those are sperm and an ova or egg. Now, sperm are produced by the male of the two biological genders, and they produce that by a process that's called meiosis. And when I think about meiosis, I think the fact it starts with me. So the idea that they are producing little me's is what I'm thinking when I think about that. So that's the process of producing what are called sex cells or gametes. Sperm for men, and for women, it is an ova or egg. Now, just to let you know, these have what we're going to talk about later is chromosomes. So they have half the number of chromosomes that we see in adult or just the formation of human cells or somatic cells, which are all the other cells besides your sex cells, which are gametes. Now, because they have half the number of chromosomes, when they come together through the process of fertilization, that is when the egg actually meets the sperm, or the sperm meets the egg, and they combine their DNA. At this point, we get what is known as a fertilized egg or zygote. That has a full number of chromosomes, or what we know as the full set of DNA. And then this is what we're going to be focusing on for this lecture is mitosis. So mitosis is a process of dividing cells to make new cells. And so you can see when we have mitosis, we have basically this individual cell making copies. And so you get growth here of basically a fetus developing. And then that will continue to grow into mature adults. And this whole process of getting bigger and changing is all based on the idea of mitosis happening. Now, mitosis is what allows us to grow. It allows to make or replace old broken cells or if we damage them to replace those cells. And so that is going on in almost all of our cells. And meiosis is when you take mature individuals and they produce those sex cells, sperm or egg in human species. And meiosis only occurs during reproduction. So mitosis is important, as I stated previously, for many roles. And so it helps organisms to grow, for repair of tissue. So if you cut yourself, as you can see with that little boy, or in some species, it helps to regenerate lost body parts, right? And so when we look at those are the main reasons where our body is helping to basically, or as you can see in this little lizard here, replacing or making new cells. Some organisms actually reproduce asexually by mitosis. And this is the idea that we're going to talk about called binary fission. So, but with cell life, or life in general, right, comes cell death. And so that is absolutely a part of life as well. And the process of cell death, or programmed cell death, is what's known as apoptosis. And I think about a cell popping to remember this. And what's so cool about this is it can actually carve out distinct structures during development. So you can see, compare the 2 feet here, and you can see that this has this webbed, or this tissue between the two, right? And so in this, apoptosis allows for those individual units to actually be separate. It's kind of what happens in the human hand and toe as well. Now, to make new cells, you can think, if we're going to make a new cell, it's kind of similar to either if we're going to buy an additional house. So, if you have the money and you have a home already, you might decide, I want to be able to spend some time in a really nice vacation home. So you decide to buy a vacation home. Well, you can't just take all the furniture that you have in your home and divide it between the 2 houses to have 2 complete sets of furniture for both houses. You'd only have both homes halfway filled. So you would have to buy all new furniture for the new home. So that's essentially what we're going to do when we make a new cell. And we're going to start making all the new stuff in terms of DNA replication. As we learned previously, cells have compartments, eukaryotic cells, and for those, they have things like mitochondria or Golgi or chloroplast. We're going to have to make new sets of those as well. But the most important thing that we can start with, though, is making a complete new set of DNA so that both cells can have the instructions to make the proteins that will become part of the individual organism. So we are going to essentially, for each cell, copy the entire genome, which is all the directions or all the DNA that every cell has. So what you'll want to do now is look in to the description below, and you will see the link for the very first video, which is an overview or introduction to DNA replication. You do not need to take any kind of notes for this, just watch it. So in DNA replication, both of the strands are copied. So if you remember anything about DNA, you will remember that DNA is made up of 2 chains of nucleotides. So what is a nucleotide? Those individual units. So if you look at this, each one of those is a nucleotide. And you can see that we have each one of these is a strand. So you have one strand here, and then you have another strand here. Now, when we go to make a copy of DNA, we're going to use what's called complementary base pairing. And this is when we take these individual nucleotides, and as you can see here, we have a T, which stands for thymine, and it will base pair with A. This always happens in DNA, unless there's a mistake. And that's something we'll talk about later. And then if you look at the next one, here is C. And if you notice, it always pairs with G. And so you can see here, this always happens too in DNA. So each strand, what we're going to see is it's going to split here. And these are going to become, this is going to become a template, and these both are going to become templates to make the new strand. So in this process, with that happening, that is what's known as semi-conservative. And if you think the idea in just the general sociological aspect, conservative means holding on to the past, right? So semi-conservative would be, okay, I'm going to hold on to some of my past, but I'm going to create something new. So that's what happens in DNA replication. As you can see, we have each strand is separated, and so each strand is going to have what's called a parent strand. And then if you look, each strand is going to have a new strand. These parent strands are what we know as the templates. So we have a template here, and we have a template that's here. And then you're going to end up making the new strands or the daughter strands here. And that's going to give us two identical DNA molecules. So how does this work? Well, there's three huge main processes. The first one is that it's going to unwind the DNA or unzip it so that you can make the two strands. The next one, you're going to essentially synthesize a new polymer of DNA by adding the complementary base pairs. And then the third part of this, you're going to actually take, because not everything makes complete strands of DNA. So the small individual strands that we have, we are going to take and put them together, which is called ligasing. Now, just a reminder, when we make this new polymer, we are going to use complementary base pairing so that we can assure that the new DNA strand has the correct sequence. So T is going to bind with A, and C is going to bind with G. Now, when we do this, when we make this new piece of DNA, it requires a ton of energy. So we had talked about how to make, or the process of cellular respiration, how to make ATP, because ATP is the chemical molecule that's required to allow enzymes to work, because these enzymes need energy. So this is going to help to make the new polymer by adding nucleotides. It's going to unwind the DNA and do all that. So energy is needed for all those processes. Now, it's not one enzyme that is going to replicate a chromosome, which is a piece of DNA, or a set of DNA, right? It's going to happen simultaneously in multiple spots. So you can see here, here, and here, where they start is called the origin of replication. And so each one of those, you're going to have an enzyme that goes on there and binds, unzips, and then adds the new strand of DNA. And so you can see it's happening here, here, and here. And so when we finally get these together, they're going to be strands that are separate. So in this last part, they're going to ligate it together so it's one large strand. So in eukaryotic cells, you do get this multiple origins of replication happening, and enzyme helping each other and dividing up the work. So what I want you to do right now is to take and actually watch this video on DNA replication and place in all the enzymes that we have that we've already talked about. You want to take notes over this, and then after we get done, I'm going to review this process in more detail than I did the first time, so that you can make sure that you kind of get the idea of what's happening. And you can find the link below. So let me move back to this so that we can talk about every single enzyme that we have. You've already been introduced to it in an overview and a review by the Amoeba sisters. So let's talk about this. So the first thing that we have here is what's called helicase. And if you remember, you can always tell what that you have an enzyme because you have the ACE at the end of that. And the beginning of it's telling you what it's going to do. So we have a helix here. And so it's going to unwind this and unzip it. So you can see right here that you have that number 1. And that is the helicase unwinding and unzipping the DNA. Now, as you realize, and we look, there is something called topoisomerase, which is going to basically hold this guy and make sure that it doesn't cause a lot of problems or fall apart. So the idea that I think, if you're looking at this, I have this cord right here. And if I have a piece of DNA, it basically winds like this, right? So see and look what happens as I unzip stuff. It becomes really tight and essentially could fall apart. So what we're going to do is that topoisomerase, which you're not going to define if you do the handout, you'll notice that I put that in there. It kind of holds it in place so the whole thing doesn't fall apart. So essentially what happens is it holds it right here. So as this is pulling apart, it protects all the DNA upstream. The next thing that we're going to do is we're going to make and synthesize a new piece of DNA. Now, for that enzyme, which is called, again, we have the ACE on the end, which is the enzyme, and it's making a new polymer. Polymer is a bigger molecule, which is what DNA is. And so they are going to use complementary base pairing to add. Now, for this enzyme to bind, it needs a recognition site. And so that is an RNA primer that is there. And when I think about primer, I think about kind of if you're going to paint a car, right? What's the very first thing that you put on for the car so that the paint has a really good paint job? And that is a primer. So it happens first and it primes this process. Essentially what it does, if I have a room of people in yellow shirts and I want everybody in that room to go and talk to one individual person to sign in, I'm not going to get that sign-in person a yellow shirt like everybody else, because then you're not going to know who to go to. But if I put someone in a red shirt in a room of yellow shirts, I can easily say, hey guys, see that red shirt person? That's who you need to go sign up with. So if you notice, this is RNA. It is a different type of nucleotide. It is not DNA, right? So it's a perfect idea that it essentially is something that was like, wait, why is there this RNA and this DNA? This doesn't look right. Oh, that tells me where I need to go as an enzyme and start. So that's the process there. And so this DNA polymerase can only walk in one direction. And so it's kind of the idea if I have two people. And when you walk, you can only walk in this forward movement, right? Like if I want to be really efficient, I'm not running backwards because I can't see, right? Say you cannot turn your head or anything. So I'm going to walk forward, right? And I have a path that you're going to walk on. So there are two paths. But what do you do if you have a person and you tell them you can walk, but you're facing the opposite direction. They can only still walk that way. So how do you get them? Do I get these two people to cover the same path? Well, what happens with this person, they just walk straight. It's really efficient, it's really easy, and they just go. This other person, though, doesn't have it that easy. They can only walk on this path in this direction. Now, they can do anything off the path, but they can only walk on the path. And as they cover the path, or as this person walks, this path will continue to generate in the new direction. So what this person can do is they can run, they can come over here, they can jump on the path, they can walk, they can go this way. So they'll cover the path here. But then more road needs to be covered. So they'll jump off of here and then go over here and run. And they'll go this way. And then they'll get off the path and they'll jump over here. And they'll run and they'll go this way. And sooner or later they'll cover the same path. But this one is always going to be a little bit slower to finish. They're both going at the same speed because they both can walk the same speed. But because this one constantly has to jump off and get back on and jump off and get back on, it can be a little bit behind. So with this both said, this is what we would call the leading strand in DNA replication. That is what's happening here. Because that enzyme, is going in the same direction as the helicase is unzipping. It's what's called the lagging strand. Because they always fall behind a little bit. And that's because this one is going in the opposite direction. So as this unzips, there's more of this DNA that becomes exposed. So he'll come off of there and then he'll bind back and go this way. Now, because he's making these small segments of DNA consistently, these small sequences are what are called Okazaki fragments. They're named after the husband-wife team that discovered them. And so we are going to make this polymer, but at the end, we have multiple origins of replication, and we have these Okazaki fragments, so we have to put them all together, which is our last enzyme that's called ligase, because they are going to ligate, which means join the strands together. So this is the whole process that's happening. So if you like, you can watch this review again to get a good idea and kind of see everything that's happening. And just to let you know, if you see on here, this primase that's there, that is what makes the RNA primers that we talked about. And again, ACE and what it does at the beginning. So that is what happens. And in eukaryotic cells, we're making new chromosomes. And in prokaryotic cells, they're replicating the DNA. And that process, instead of being called just basic DNA replication, or the process of the cell cycle mitosis, which we'll talk about next, is called binary fission. This happens in the domain bacteria and archaea, and it's asexual reproduction. That is just taking the original cell, and if you see here, that is the original DNA, and you can see you make a copy, and then the copy goes over here. The original is here, and then all of a sudden you have two exact copies. Now this is really fast. It doesn't take as much energy as everything else, and the problem with this is you can replicate organisms rather quickly, but everything is exactly identical to the original parent cell. So whatever kills the parent cell will kill the daughter cells. Now, prokaryotes can get new DNA in other ways too. In both bacteria and archaea, you can have a donor cell, which is the original source of DNA, and these are what the small circles are called plasmid, and they can transfer a gene to a recipient cell using what's known as a sexual pelus. And this is just that connection that happens there. So you can they can take their genes and transfer them to a new recipient gene cell to allow them DNA that's going to make them more robust. Healthy cells, bacteria cells, and archaea cells can also absorb DNA from dead and dying cells. They can incorporate them. So those cells will release fragments of DNA, and they will essentially bring them in and incorporate those new genes into the actual live bacterial cell. And so once we make the new piece of DNA, what's so interesting is that then that DNA will wind up extra tight and condense. So you can see this is what DNA looks like before condensing. And this is a form that's called chromatin. And I think normal. That's what it's like when it's not doing anything special. But when it goes to condense, here, you're going to see it's going to condense into chromosomes that you can see. Those are when we look at the little X's. And each side of that is going to be what are called sister chromatids. And I think D for divide, because that's what these are going to do. They're putting in a form that's really easy. It's kind of like taking all your stuff in your house when you go to move and you condense it into boxes to make it easy to move it around so nothing gets destroyed. That's what we're doing here. And they coil it tightly, almost like coiled yarn. And it goes from chromatin to chromatids. And that's when the cell will get ready to divide. And that will lead us to our next topic of cellular division, which is the cell cycle and mitosis. And you can watch the next video to cover that.